Humans have been manipulating the genetic code of organisms for millennia by selectively breeding individuals with desirable features. As scientists have learned more about how to read and manipulate code, they have begun to take genetic information with useful features from one organism and add it to another, creating the basis of genetic engineering. More recent advances have allowed scientists to make new sequences of DNA from scratch. Recently, scientists have combined these advances with engineering principles to design organisms to do new things, like produce biofuels and pharmaceutical products. This is the essence of the new exciting field of synthetic biology. In summation, synthetic biology can be defined as the assembly of new biological parts and devices and systems, and the reconfiguration of existing biological systems for useful purposes. An underlying goal of synthetic biology is to make the process of designing, building, and testing biological systems easier. To meet this goal, scientists have been developing methods that support the design and construction of multi-part synthetic biological systems from standardized biological parts. The first widely accepted standard for biological parts was proposed by Tom Knight in 2003. Knight proposed the BioBricks standard for assembly of genetic parts. BioBricks are standardized DNA sequences that can be used to design and assemble larger synthetic biological circuits from individual parts. These biological circuits can then be incorporated into living cells such as E. coli to construct new biological systems. Examples of BioBrick parts are promoters, protein coding sequences, inverters, ribosomal binding sites, terminators, and plasmid backbones.
BioBricks are readily found on common source sites such as the Registry of Standard Biological Parts and BioFab. The Registry of Standard Biological Parts was designed by researchers at MIT, Harvard, and UCSF. The registry contains information and characterization data on a variety of BioBrick parts. The catalog describes the function, performance, and design of the parts. Each BioBrick comes with a unique identification code. All BioBrick parts are flanked by a prefix and a suffix. These sequences make it possible to put together different parts to create biological circuits. There are many different standards used to join parts together that differ mainly in the restriction sites incorporated into the prefix and suffix sequences. The assembly techniques previously described using traditional cloning with restriction enzymes are efficient and simple to use. However, they leave recombination site sequences, or SCARs, in the final construction that add 8 to 13 extra amino acids to the protein of interest. Golden Gate cloning circumvents this problem.
Cloning is a simple, rapid subcloning strategy that is used to transfer any DNA fragment from an entry clone into an expression vector without leaving recombination site sequences, or SCARs. In traditional cloning, restriction enzymes bind and cut at exactly the same spot, so only one type of sticky end can be produced. Golden Gate cloning overcomes this by taking advantage of the type 2S endonucleases, whose recognition sites are distal from their cut sites. Their recognition sites are not palindromic and therefore are directional. Type 2S endonucleases create 4 base pair sticky ends next to their binding sites, regardless of the adjacent nucleotide sequences. If a part is flanked by 2 binding sites for type 2S restriction endonucleases, digestion will lead to predefined sticky ends at each end of the part. If a destination vector is added that contains specific type 2S restriction sites, its intermediate piece gets replaced by the assembled parts. PCR can be used to incorporate complementary sequences next to the type 2S recognition sites for the linearized vector and the parts. After PCR amplification of the vector and fragments, Golden Gate cloning can be performed in a single tube reaction with all DNA parts, restriction endonucleases, and ligases. After transformation, the antibiotic resistance of the destination vector selects for the right clones. Methods have been proposed to introduce type 2S endonuclease sites into plasmid backbones to assemble BioBrick parts together using Golden Gate cloning. Assembly using Golden Gate cloning is advantageous over traditional cloning, as there will no longer be SCAR sequences between joined BioBrick parts. These methods are further described on our website.
Current BioBrick research is mainly focused on developing and expanding upon the assembly standards and techniques already proposed. These new standards address several challenges faced by engineers, such as lack of efficient tools for fine-tuning gene expression and constructing pathways with multiple gene components. BioBricks are also being integrated into assay design by using BioBricks systems in yeast to create renewable wholesale biosensors. In the future, BioBricks will be integrated in the development of metabolic pathway construction. Research will continue to focus on optimizing DNA assembly techniques and constructing new BioBrick parts and vectors. Golden Gate cloning has been incorporated into many different research areas. For example, CRISPR technology has adopted Golden Gate cloning to insert oligonucleotides specifying a gRNA target sequence into Cas9 containing plasmids. Golden Gate cloning is also being used with standardized parts to construct multi-gene constructs for plant transformations. In the future, it is likely that Golden Gate cloning will be used in other research areas and will become more popular than traditional cloning techniques. iGEM is an annual synthetic biology competition aimed at undergraduates and high school students. Teams are given a kit of biological parts, and they use these parts to build biological systems that can operate in living cells. One example of an iGEM project uses BioBricks to address a real-world environmental concern. Students at the University of Pavia, Italy, designed a biological system to convert lactose from whey, a cheese byproduct, into ethanol to be used as a biofuel. They added BioBricks containing protein coding sequences for enzymes such as beta-galactosidase to allow E. coli to metabolize lactose into ethanol. For more information on BioBricks and Golden Gate cloning, please check out our website, linked in the description below. Press that thumbs up and leave a comment. What will you build with BioBricks?